Yeah, sure. We can get started.
I think we can start again.
Yeah. So we have nearly 25 people today for this event. Welcome everybody. So this is a basic science core of DCC FAR open house. Typically, a few times in a year, we organize this open house to kind of reorient our members. What kind of technologies are available for them to enhance their research on HIV, and also what we offer them. So every time we focus on certain aspects, or this time we are focusing on what kind of flow cytometry facilities are available for us. So we are kind of a citywide CFAR. So everyone is working at different institutes. And so we try to kind of decentralize our services. So today there will be three speakers. They will talk about the flow cytometry facilities and how you can utilize those, and what are the applications and everything. They will talk about it. Greg from GW, Karen from Georgetown, and Namita from Harvard University. So these are the speakers. This is a 1 hour event. So let's get started.
Okay, so I will introduce the first speaker, Dr. Greg Creswell. Dr. Creswell did his bachelor's at Southern Illinois University, and then he do a thesis. So he went to do his. What is your thesis? Sorry, I was prepared, but I guess I wasn't. Purdue University, sorry, where he studied the role of the folate receptor beta as a marker of immunosuppressive myeloid-derived suppressor cells and tumor-associated macrophages in the tumor microenvironment. After that, he actually was at Purdue University in the flow cytometry and cell separation facility as a teaching assistant, and then as a postdoctoral researcher again at Purdue University. So Greg joined GW last year. Sorry, Greg, that's and he actually, he was part of his search committee, and he was really excellent, the best candidate that we have. He not only, he has a lot of experience with flow cytometry. Has a PhD in immunology, and so that also helps, right, with when understanding the scientific questions that people wanted to work with more than 10 years of research experience in different academic labs. It is expert in regulatory myeloid cells, multiparametric flow cytometry, live cell sorting, as well as many other skills such as single-cell RNA-seq platforms and things like that. So with that, I just want to let Greg introduce the GW flow core. Greg, go ahead, and I think you can slide.
That introduction. I'll go ahead and share my PowerPoint here. All right, can everybody see that?
Yes.
Good. Move everybody's little window out of the way here. So as we said, I'm the new, well, it's not so new, it's been a year now, GW Flow Cytometry Core Facility Manager. I'm under the GW Cancer Center, but we obviously service the entire GW campus, and we have options for external users, which I'll discuss a little bit as part of my presentation. So if you have an overview of the GW Flow Core, we have analysis and sorting services. So we have live cell sorters and conventional and spectral analyzers. I provide training and guidance on experimental design and equipment use. So this is part of my immunology and PhD experience. So I try to kind of help with experimental planning, troubleshooting, panel development, kind of a start to finish approach for groups. We offer assisted or independent use of our instruments through training. We provide software access to core users through FCS Express or FlowJo, and I provide training and analysis assistance on both those platforms. And then I have one cell sorter that gives you the option for independent use, so users can be trained to sort on their own, or we can do live cell sorting as an assisted service through me. And so just kind of an overview of our equipment that we have available. Our conventional flow analyzer is a BD Celesta. This is a 12 color instrument in a 3 laser configuration, red, blue, and violet. This one is available for independent or assisted use. We have an assisted rate at 80 an hour or 45 an hour unassisted. I have some more information about its configuration set up through the GW FlowCore website. This is kind of our standard flow cytometers. This is what most people are going to be familiar with or have seen before. Kind of moving up a step in sophistication, we have a Cytec Aurora spectral analyzer in a 3 laser configuration. This is going to give you closer to 24 color panels comfortably. Realistically, the instrument sits between about 20 to 25 colors. It is a 34 detector instrument, so there's always a little bit of flexibility in there depending on what you want to set up. This is an independent or assisted use instrument as well, and it has the same fee structure as the Celesta. So for basically the same amount within the core, you're able to do high parameter analysis using this instrument. So this is one that we can start to build Tisney plots and do some association work with much larger color panels and very broad analysis of groups, or we can do very highly focused phenotypic analysis of subpopulations of immune cells with this instrument. For cell sorting, we have a BD Influx. This is a stream-air, kind of a high-speed sorter. It's a 15-color configuration on 4 lasers. It will do 6 populations simultaneously or 96-well index sorting. This one is assisted use only due to the complexity of the instrument, but it's got a pretty broad range of nozzle options available. So this allows us to sort small cells very quickly at the 70 micron, or we can sort very large fragile cells at this end of the spectrum. And we do have options for small particle detection. This instrument can do some like bacterial sized options. There's some proof of concept for exosome work with it, which is pretty impressive for being a stream-air sorter. And this one is 125 an hour. And again, I have more information on all these instruments through the website as far as more detailed information on their configurations, filter sets.
Greg, can I ask you a quick question?
Yeah, sure. Feel free to interrupt.
Any standard, yeah, any standard experiment, how many hours a person can expect for sorting?
On this one, it's pretty quick. So right now I've been averaging on this instrument about 1 to 2 hours.
Okay. So in terms of the cost wise, I want to kind of tell them that it will not be so expensive.
No, this one isn't too bad for the user. Most of the cost on this one is associated with the setup time. This one's pretty labor intensive on my end. For the end users, it's not bad. You can show up with samples. I do the setup, I do the sorting. Most groups are through 2 hours is a longer sort. A lot of groups are averaging about an hour to an hour and a half.
Thank you.
Sure.
Do you have a different price scale for the outside and inside users?
Yes. Yeah, I'll show the external rates. I have kind of a summary slide at the end where we can discuss that.
Okay, thank you.
Sure. So continuing with our live cell sorters, we have a Sony, an SH800Z. So this is a 6-color instrument, so it's a little bit more scaled back from the Influx as far as what kind of panels you can run. This one only does 2-way sorting or 96-well index sorting, and it is limited on its nozzle size. So this instrument is a little bit slower. The advantage to this one is it is chip-based with an automatic setup. So this one can be used independently. I can train users to do their own sorting on this instrument. This one's kind of recently absorbed into the core. So right now we have the assisted rate, and then we're working on an independent rate with finance at the university. So I'm obviously advocating for a lower independent rate since the instrument does set up, and I'm actually hoping to reduce this 125 a little bit. Most of this was based on sorting on the Influx, which this instrument is not as difficult to set up and operate. So I have a few users now that we're starting to transition to independent use on this instrument, which is nice, kind of expands our sorting capacity.
Greg, since this one is chip-based, who is responsible for the chips?
Right now, I've been providing the chips as part of a service of the core, especially in light of the current fee rate for this instrument.
Okay.
And I also provide the setup beads that the instrument needs to run its initial configuration. And then again, as a core service, we do provide site licenses for FlowJo and SCS Express. So any labs that use the core have access to one seat, potentially on both platforms if they want it, as a core service. And then additional seats, if you have multiple students or people that want access, I sell those at the reduced kind of bulk site license rate the core pays. So we try to provide broad software access for everybody, since that's a big part of the analysis component. And then experimental design.
Can I ask you a question?
Yeah, sure.
So I was wondering, are all these instruments that you are noting can be used with infectious samples, like this sorter that users can, it's like a self-service?
Yeah, so I didn't explicitly point it out, but the BD Influx is in a biobubble configuration as part of a BSL2 plus lab. And the Sony sorter is in a biosafety cabinet. So both sorters can be used with infectious material. Ideally, the Sony sorter has a little bit better setup since it's in a dedicated cabinet as opposed to the biobubble, but both are configured for infectious material.
Okay, thank you.
Sure. And then we do have a core service where we will just take samples, run them, and analyze them. So that's the instrumentation rate, and then data analysis is about 95 an hour. For most groups, it takes about a half hour to an hour, depending on complexity of their data. But this is a complete service, essentially. Groups can bring me samples, I run them, analyze them, send data back. We've been trying to offer this for groups that are running kind of more limited studies. They have something that was part of a grant or a review. They just need it done. I can facilitate kind of a start to finish to get things done quickly for groups. As far as experimental design and troubleshooting of experiments, that's all part of the equipment use. I try to get everybody through their studies. I did set up a fluorofinder page for all of our equipment. So you can go in, put in your antibody panels, and fluorofinder will be specific to the instruments that I have in the core. So you'll be able to build kind of a complete antigen and flow panel using this website. Kind of streamlines the process a little bit for users. Right now, all of my training is in person for equipment. So that'll be analysis and sorting. For the data analysis, software training, or experimental design work, we obviously have virtual options for that. Everything is arranged right now via email to me. And then we have access for internal or external users. And kind of a summary of that fee structure, specifically for external users, nonprofits are a 59.5% increase of our internal rate as determined by finance. I like the nice specific number that they gave me. And then for-profit entities, external users, that is twice the internal rate. And then internal rates are campus-wide for the GW system.
I mean, I think that external, that is our IDCs or grants. I think it's like the same indirect cost that we charge in the grants.
Yeah, I think that's what they did.
I think it's something similar.
It always gets a chuckle out of people when I tell them this like hyper specific number, but.
So Greg,
yeah, sure.
Is there any cost associated with the training?
So generally for the training, I am just charging the assisted instrument rate. Typically training for the Celeste and the Aurora is a couple hours for kind of the introductory training. And then there's usually maybe 1 to 2 sessions after that where I'm just sort of observing the user to make sure they're comfortable. And then they're allowed to move to independent use kind of during business hours while I'm around in case they have questions. So I try to get people through that assisted rate as quickly as the user is comfortable.
Mm-hmm.
And I'm happy to take any questions or if anybody has more specific questions about like kind of stuff we can run or.
Any new graduate students in the audience? They can ask questions. Alan, you were saying something.
I have a question for the core directors. How does this all relate to the Fortessa and the cell sorting that we have through the CFAR?
The Fortessa, I mean, the fee, I mean, there is no fee per service because we cannot have, so it's people using, and then we, the groups that use it, they pay some of the expenses that are associated with that. But we don't, unless it's in Georgetown, but we at GW, we don't have another sorting capacity. So as a part of the instruments on the DCC far, we only have that Fortessa, right? I don't think that there is sorting.
Yes, there is no. Yeah.
So
Thank you.
No problem.
Thanks, Greg.
Sure.
There's a good question in the chat about how it works.
Oh, you see the chat.
Oh, there's one in chat.
So next is the Georgetown, right?
Yes.
Marta, are you presenting? You're going to the present, right? I think you're muted.
No, I am. Karen.
Yeah, Karen, yeah.
Karen,
But I think
Sorry. And
get
mine actually is more theoretical.
okay.
Because I was unsure like who the audience was and what should you do. So basically, we're very similar to Greg in the standard flow cytometry things, information. But we additionally have a mass cytometer, which was a thing that I was going to talk to you about today. So this is I just have the contact information here. At Georgetown campus, Georgetown University, we are part of the Lombardi Shared Resource, but also university-wide and also work with other universities. And we have 3 flow cytometers, a small particle analyzer and a mass cytometer. So for flow cytometers, which is what Greg was talking about, we have a Faxaria for a sorter, also in a biobubble, which can do infectious samples. And then we have 2 analyzers, which are Fortessas. Similar configurations to what he has. We don't have a spectral analyzer at this time. We're working on getting one. We also have a nanosite for a small particle analysis, which is available for users. All the pricing is on the website. I didn't know you wanted like specific pricing and stuff like that. The thing that I wanted to talk to you about was that we now have a mass cytometer, and we're specifically what the flow shared resource works with is suspension mass cytometry. The instrumentation we have also can do imaging mass cytometry, where it can be slide based and you can of the different antibodies on a slide based system. But the flow cytometry core is not in charge of that. If you want information about that, I could give you the name of the person that's the contact for that. But I'm going to talk just about the suspension mass cytometry. So the thing about suspension mass cytometry, since somebody asked about theoretical, for conventional flow cytometry, you have fluorochromes conjugated to antibodies. So for mass cytometry, instead of fluorochromes, you have a metal conjugated to it. to the antibody. And the thing about this is that unlike flow cytometry, where you have different fluorochromes and they have different emission wavelengths and they overlap. I don't know if you've done any flow cytometry, you know about compensation and overlap and stuff like that. With the mass cytometry, they don't overlap. So they're very pure. So you can do a lot of different metals and differentiate them with very minimal overlap. And you can do more than 50 metals at a time. So you can get, do a large analysis with a bunch of metals with, not having to worry about compensation a lot with this. So that's what the mass cytometry brings to the table. It's very similar to conventional flow cytometry in this, staining of the samples and the preparation of the samples is very similar to it. And this is just showing that, there's a nice sweet spot of isotopes that are used for most of the mass cytometry experiments. And then you can also do like live dead, like you can do in conventional flow cytometry. And one thing that we've particularly found exciting is like to do barcoding, where you can actually combine a bunch of samples together and do all the staining and the preparation together, and then debarcode it, and you can analyze multiple samples from one tube. So basically I just wanted to tell you, you have a cell suspension and it goes through a nebulizer, and it's sprayed into a heated chamber, which ionizes the cell. It strips the lower molecular weight, it strips the lower molecular weight particles off there, like carbon and things like that. And then what you end up with is the high heavy metals are left. These are flung through a chamber, which is why it's called CyTOF, time of flight. And the time that it takes to go across the chamber correlates to the molecular weight of the actual metal. So that way you could tell which metal it is, which is basically which antibody it is. And it's done kind of in little bursts and little pushes, and that's how you can basically tell the cell. So you don't have forward and side scatter like you have in conventional flow cytometry, but you have these pushes that come through, and then it pretty much comes out. The results are very similar to basic flow cytometry. You can use the same analysis programs that you can use for flow cytometry, except usually, really, you wouldn't want to do the mass cytometry for small number of markers. You'd want to do this for a larger marker, maybe 15 or more markers, because that's the beauty of it, is to be able to do a lot of markers at the same time. And this is just talking about how it's different pushes through there. And then this is basically what the data looks like when it comes off of it. This is called a rain plot, where you can see these little pushes, and each of those basically is a cell, and then you would see which of the metals it has in that push and the intensity of the metal in that push. It's just like flow cytometry, where you can see not only whether it's present or not present, but the intensity level of it. So it's very similar to flow cytometry. It's just that you can do a lot more parameters at the same time. And then you can analyze it with like any tools that you do flow cytometry data with. This, I'll talk about that later. And you can see there's been comparisons with regular flow cytometry, and it's very, results will come where it's comparable to regular flow cytometric data. Except it doesn't have as much spreading problem as it does in regular flow cytometry data. So it's actually a little cleaner than flow cytometry data, and you don't have the overlap problems with it. This is a picture of the current flow one that we have. You can see the nebulizer there and going into the chamber. But we are in the process of installing a new one called the XT, which is the newer model of it, which it actually has an improvement over this and has a like a carousel for multiple samples. This thing, the Hyperion, is the imaging system, which can be added onto it, which we do have this Hyperion. If you wanted to do imaging mass cytometry, this is the instrument. It's called the Hyperion. As with
Karen,
yes,
is this similar to image stream? The equipment that is kind of has a sort of a confocal microscopy coupled with the flow cytometer. This Hyperion. So what do you mean by imaging?
so basically what it does is you put the slide in there and it will ablate a cubic millimeter of tissue and analyze what metals are in that tissue, and then basically make a map of, you know, what things are there. So you select your region of interest and then just do your region of interest. Most people do like a tissue array on a slide, and then they'll have a bunch of regions of interest on the slide where they'll look at the different, so you can see like the locations of different cells, you know, in context of each other. But it's basically using the mass cytometry.
Thank you.
That does take a lot longer to do, to ablate the tissue like that. So, I mean, those experiments, so we've been sharing this instrument, which is this Hyperion goes onto the Helios, you can see there. That's why we're now getting an XT, which will only be for the suspension, but the Hyperion, that they run that like all weekend. It takes a lot of hours to do the imaging. So, but for the suspension, it doesn't, you know, it does not take that long to run the samples for the suspension one. This is just to show you that CyTOF really is kind of like the wave of the future, where the number of publications and the clinical trials using CyTOF, I'm working right now on 4 clinical trials utilizing the CyTOF to generate, you know, data for patient data at this time. And so it really is becoming, and I've heard people say that, you know, if you have this in your grants, the grants are more successful in getting funded if you're having the CyTOF technology in there. It's kind of the wave of the future. Here it is in clinical trials. You can see that a lot of clinical trials are using it. The one thing that I wanted to talk to really quickly about was, it's just saying it's a tier 1 assay now. Mass cytometry. The thing I wanted to talk to you about was this, the MaxPAR Direct Immune Profiling System. So we do both mouse and human at this time. We haven't done any other species. The way that this was developed by Fluidigm, they concentrated mostly on human. So they have a lot more reagents for human, but they do have human and mouse reagents. And if they don't have the reagents, we here have been conjugating the antibodies for people to the metals, you know, to help them out with their experiments. But for human, they have this thing called the MaxPAR Direct Immune Profiling System, which basically is a tube that already has 30 markers lyophilized in the tube. And you can put 300 microliters of blood or 3 million PBMCs into this tube, and that there's a software, the Path Setter software that I showed you previously, that will actually do an analysis on all of your cell types within the tube. And it's a very standardized system. And like a lot of the clinical trials, this is what they're using. So you can, it's very convenient because, you know, the cells can be shipped on wet ice. Like, you know, if you weren't, like, we have Georgetown has a location in Hackensack. They can just ship samples on wet ice overnight, and it doesn't interfere with the assay, which has been tested by multiple labs to make sure that everything is standardized for this use. So this is a very standardized assay if you just wanted to, you know, have always exactly the same standard panel for your tests. And basically it gives you all of the regular, all of your cell types. I don't know if the thing, okay, this tells you what's in the panel right now. And they also have expansion kits that you can add to this, which would, you know, for different specialties. Like if you wanted a T helper add-on kit, it has some other antibodies that would complement the 30 marker basic panel. And so this is only human. For mouse, the biggest one they sell for mouse studies is a spleen lymph node 16 marker kit, which, you know, sometimes we use it as a basis to build other things off of it, or else, you know, we'll build a whole new panel for you to do your experiments. So it's slightly more expensive. It's more expensive to get started because to buy all the antibodies, because if you're going to be doing an experiment like with 50 parameters, then you have to buy the 50 antibodies or whatever to begin with. So it's the initial startup charge is kind of expensive, but once you get going, it's comparable to flow cytometry, the pricing per sample. And I'm here to help you with anything that you want to do. If you want to design a panel or, you know, conjugate some antibodies. So basically we do a lot with BioLegend because they have reasonably priced antibodies. You can buy non-conjugated antibodies from BioLegend and we can conjugate it. And, you know, we have the conjugation kits already. And we're pretty much selling those for the price that we're paying for them, you know, without making a profit off of it. So there's a lot that can be done with this region of interest. And if you have very small samples, you know, that's okay because you can like do the barcoding like I was talking about, and you can basically label which cells are coming from each sample and mix them all together and then do staining and get some nice results from that. And so that, the max part, the MDIPA, we do that quite a bit for people. But we do, it depends, whatever people want from their project, we do. And like I said, they have these expansion panels here where, you know, you would look at your panel, like what you want to add it to, you know, depending on what you're exactly focusing on, of which panel you could add on to. And like I said, you only need 3 million PBMCs or 300 microliters of blood to do the test. And so this is just showing the different colors and the expansion panels. Like the orange ones are the basic panel, and then the add-on ones are in the purple. So so there is a lot that can be done, and we're here to help you with any of it. or your regular flow cytometry needs. Does anybody have any questions?
I do have a question.
Yes.
Does with CyTOF, can you do intracellular proteins, or is
Yes.
only for surface stainings? Because
Yes.
I only see, like, surface markers.
Yes, the MDIPA, the immune profiling, the original panel was all surface, but actually some of these expansion panels do have intracellular. And right now we're doing, it's not the MDIPA, which is this one, but they have some kits, and we're now working on a kit like a T-reg kit. So, we're doing the FOXP3, and then the people wanted to add like Helios and different things like that. So we are working on some T-reg kits now, which are, have intracellular staining. And, we've tested out a bunch of different fixed perm methods to try to get like the best signals from it. So yes, you can do intracellular.
And it allows you to do surface and intracellular.
Yes, yes.
Okay.
It's just like flow. You would do
Yeah,
your
okay.
surface markers, you do your fixed. Yeah, there's a couple extra steps. Like at the end, you label everything with iridium, basically just to identify the cells, because you don't have your forward side scatter anymore. So that's just like an extra step at the end. In the beginning, you do your live dead. It's very similar to flow. Yes, but as you see, since it vaporizes the cell, you cannot sort with it. So because you vaporize your cell.
And sorry, I don't want to take all the questions, but if you were going to say what is the limit of detection and the number of cells that you can, of the events that it will be a cell, for example, can you detect one cell in a million that it has a specific phenotype?
Well, you would just have to collect a bigger file to, you know what I mean? Like
Yeah.
the rarer the event, the larger file you have to collect. Right?
Mhm.
So, yeah.
So, Karen, how much is the wait time?
What do you mean by wait?
Wait time in the sense, if I want to use the CyTOF, is there a waiting on that?
No.
It can
If
be
you wanted to
done.
do an MDIPA, if you brought cells today, we could do it tomorrow.
Oh, okay. Good to know.
Because
Thank
usually
you.
you do the staining, and then the last step is 1 hour or overnight in iridium. So we usually do the overnight in iridium, and then we run them the next day. So, but no, we don't have a wait time, and we do have MDIPA, and we have the human T-reg panel, and we have the mouse spleen lymph node panel. We have all those in-house, and we sell them like per antibody, like whatever, at the price that we bought them. Well, we can sell you the reagents. So.
Yeah.
So.
I was going to ask, Karen, so for the quantification, right? So it's basically based on the MS, right? MS quantification of the heavy ions, right? Or because some of the like we've been using Bioplex, right, which is based on antibodies as well, but it's mostly like suspension, like cytokine detection, this kind of things. And each antibody has its own kind of standard curve, which is sometimes very peculiar, I think. So I was wondering how in your case, because you have different antibodies, how would you account for these antibodies, being not non-equal, and how would this play in quantification of your signal?
What do you mean the antibodies are not equal?
Like
the intensity?
What I mean is that, yeah, so if you look at the standard curve for, like, for example, if you look at the different cytokines, so
right.
each standard curve is very different, like for interleukin-1, interleukin-6, for example, interleukin-12, whatever. Yeah. So I think it's based on not only on the amount of the detection, like in your case, it's like it's heavy atoms, right? But also how antibodies will recognize the antigen. I think I don't know. I mean, I think it really, I mean, all these approaches really requires like very good antibodies, like highly tested and this kind of thing. So,
This is
So-
more similar to like regular flow cytometry, where when you have things that are a dimmer antibody or a lower percent, that you use the ones, like I told you, there was a sweet spot of nice masses that gave, lower signals, and then things like CD45, CD3, CD4, things that are in high abundance, you could use, not so great of a, mass. You can use like cadmiums, which are a lower mass,
Okay.
or things like that. So it is in the panel design, in the panel design, you like try to figure out what is the intensity of the signal that you're expecting to get. Is it going to be a dim marker, or is it going to be, a strong marker? And then you can select the metal based on that. Just like flow cytometry, you do that in flow cytometry too.
Okay.
And, fluorescence flow cytometry, yes.
Thank you.
So, did I answer your question?
I think I understand. So I think you're basically more like flow procedure
Yeah.
with some extended
Yeah.
probability.
Yes.
And then in suspension area, you detect like small molecules, not necessarily cells. I mean, you can do cells as well, but it's mostly based on small molecules. So you have to build some kind of calibration curves, which is a little bit different, I think. All right. Okay. Thank you.
Okay. Anybody else? So all the information is on our website, or you can just contact me if you have any questions about it. And we're ready to do your samples whenever you want.
Thank you.
Okay,
Thank you.
thanks.
Right. I think I'm going to introduce last speaker. So this is Dr. Namita Kumari.
Stop.
Yep. Okay. Yeah. So Namita, she joined my lab after being postdoc at Children's Hospital. So she received her master degree from the University of Baroda, and then she got her master of philosophy from Delhi University, and then her PhD from Baroda University. So she worked at Children's on the muscle dystrophy before joining my lab, where she became involved in the variety of the projects related to HIV. I think we started with the development of the induced pluripotent stem cells, eventually developing HIV-resistant macrophages, and then she worked on the small molecules, curcumin analogs as HIV inhibitors, iron chelators as HIV inhibitors. And most recently, she's been kind of key person studying the innate immune response in sickle cell disease as it repels basically HIV infection. So she's been senior postdoc, and then she became assistant research professor in 2017. And from this year, she's actually tenure track faculty at the Department of Microbiology at Harvard. Okay, so please, yeah, Namita, you have the floor.
I started sharing, and then let me share the screen again. Okay, so share. Okay, so everyone can see the full screen?
Yes.
Okay, so a very good afternoon to all the members and the participants of this flow cytometry workshop. At the very beginning, let me admit that we are kind of a very young member of this flow club, and we are still evolving. For the last 10 years, we had BD fax versa with 3 lasers, only flow cytometry. And this is the the company is kind of pulling the support. It will go off support soon, but till the time we have it, we are running it. We are in the process of acquiring a new flow. It's in the final stages, that is Cytoflex S flow analyzer, again an analyzer, not a sorter. And this will have 4 lasers. And this we are doing with the aim of supporting DCCFAR and the Harvard investigators. Here I have listed few applications which might be of interest to the new users and for that matter to students also. So with the flow cytometer or the sorter, what we can do is look for immune marker or cytokine receptors or differentiation markers in blood lymphocytes. We can look, detect the expression of the receptor as well as the ligands in the cells. We can analyze DNA for apoptotic cell death, cell cycle, or chromosome polydy. For the cell functions, we can look at oxidative stress, intracellular pH, mitochondrial membrane potential, and reporter gene expression, intracellular calcium, intracellular cytokine expression, and we can also look at antibiotic susceptibility. At Harvard, we have been using the flow analyzer for a couple of projects. That includes analyzing P24 expression, CD4 expression in HIV-infected cells, which are drug-treated or untreated. And we are also looking at different restriction factors expressed by cells for restricting HIV. We are looking at delivery of antiviral drug and its effect on the HIV replication, and then delivery of nanoparticle mediated anti-HIV drug, and then also the injury caused by HIV to heart and lung, which leads to neuro-AIDS and lung complications. Then adenovirus-based vaccine development also is one of the projects. And then cell cycle of tumor cell line after drug treatment, or cell cycle of cell cycle when gene expressed in tumor cells is suppressed. So all kinds of cell cycle and protein expression and these stuff that we are analyzing. And the couple of good publications also we have got, and we have been able to help investigators at the Harvard University. For the beginners, what I would suggest, what I can suggest is, when they bring in the samples, it should be a single cell suspension. And if it is from a solid tissue, they might need to dissociate it well, and they may need to remove irrelevant cell population by density gradient. If it is an adherent cell line, they would require detachment from cell culture dish and dissociation, so that the cell again is a single cell suspension and the aggregates are removed. Then after the cell suspension, you need to assess for its viability. For staining the proteins intracellular or on the surface, you need, you need to know the configuration of the instrument that we will let you know once you approach it in advance. And then you need to choose the antibody and the fluorophore depending on the expression of those intracellular protein or the receptors. So if it is an abundant protein, you might need a less a dim fluorochrome, or if it is a less expressing protein, you might need a very bright fluorophore. Then you need to standardize the blocking and titration of antibodies for optimal signal. At core, as I said, we are evolving, and as the machine is acquired, on demand, we can arrange for a presentation from the company, or we would be hiring an expert, someone to operate the machine also full time, so that any kind of support can be given. And once you approach us, you will come to know about the instrument, laser, other information, and basic flow operation supply will be available. Your antibody and all, you need to either consult with us or decide on your own, and then discuss with us the staining procedure, or if you know it well, fine, you can do stain them and bring it to us and we can analyze it, or you can yourself use the flow. And for the initial user, we will be giving training for operation and maintenance. And as I said, any kind of presentation if you need before starting, it can be arranged. For right now, for contacting or scheduling your use of the machine, this website is you can fill up the request form here, or contact Dr. Nikhai, and he would direct you how to go about it. And that is it. As I said, we are still evolving, and let's see how it goes from here. So thank you very much, and I would invite any questions if you have. It was a very basic presentation, but that's what I thought, we are as such evolving, and, not so very, we are still evolving. Yeah. Thank you.
Thank you, Namita.
Namita, also, do you know when do you think that the instrument will be at Howard University?
Very soon. We have already placed the order. As Sergey can answer that better, but
Yeah.
I
No,
understand.
I think the contract was, signed yesterday, So it was shipped, it was sent back to GW. Once it's approved on your side, then we can start putting the order in. December, we should be able to get the machine. Hopefully by the end of the year it will be installed. Yeah.
And there are already some projects waiting to be done, even though we have a very basic facility, but we do use it optimally. Yeah.
Yeah.
Any other questions? I'm particularly looking for the questions from graduate students. So earlier, Victor posted one question on the chat, and he was saying that I was expecting a theoretical framework to how the instrument works. I believe, Namita gave a very basic idea how the flow cytometer works, and I hope that answered your question. Karen also gave some basic technology, how it works for the site off. So did that answer your question, Victor? All right, so
but I think we can do more detailed kind of like presentation on the principles of in the future, not right now, but
yeah, that's a good idea.
It's nice to keep this in mind. Yeah.
Okay, I think
I agree with that.
That'll work for me. Thank you.
Great. So let me summarize. So we have access to multiple different flow cytometers at 3 different institutes, and they range from 24 to 4 color, meaning that at one time in one sample set, you can analyze 24 markers, or limited markers. You can also sort the cells from the whole blood or from the tissue suspension, a single cell suspension. One thing I would like to highlight more is the nanosite that is available at Georgetown. Karen talked about it. That machine can be used for quantitation of exosomes. Exosomes are nano-sized little cellular fragments or particles released from the cells, and you can analyze those in the biological fluids. Larger extracellular vesicles in micron size, like microparticles, can be analyzed by flow cytometry. Any of these flow cytometers can work for that. So you have access to the site of nanosite and multiple sorters and cell analyzers, and they can be used for HIV-infected cells, cell lines, or the tissue cell suspensions. And you can analyze for the infectivity in those cell population by HIV, or the activation status of the cells. The very kind of a rare population, very low number of cells can be detected in that sample by flow cytometer, and you don't have to purify those cells. So this is a very, very brief summary of what we have here. So to this end, if no one has any questions, thank you so much to the speakers. It's amazing. Really, you gave us good information and better idea of what is available and how we can access those facilities at these 3 different institutes. Thank you so much. Have a wonderful day.
Thank you.